Just want to do a quick introduction to DrugMatrix. Prime everyone to get you going on how to use it. First of all, it only works in Internet Explorer or IE. When you're using IE10, you have to go into the compatibility, change the compatibility settings, and so you expose the menu bar, and then you go select Tools, and then Compatibility, View Settings, and it's going to be an NIH.gov site. So you've already added it. I've already added it. If it's not, it'll be up here. You press Add, it'll move down, and then after that, you can launch DrugMatrix here. So right there, and it'll pop up, look like this, and then all you do is log in here. And it'll load. And so there's a basic layout to the interface. There is the search function, so if you wanted to search within different data domains, so there's a whole bunch of different data domains. There's a gene domain, a compound assay, you get the idea. And the terms you search in a given domain are specific to that domain. So if you want to search a chemical, you go to compound. If you'd want to search for a histopathology that was called in this database, you would go here. So we'll just give it a quick search here, and so I put in macrovesicular, and you'll see here lipid accumulation. We have central lobular, midzonal, non-zonal, periportal, and you can click here and it'll load. It'll take a second, but it tells you all the studies that were done or the experiments that were done where histopathology was evaluated for this endpoint. And we have first one here is carbon tetrachloride, 7 days, 1,175 milligrams per kilogram. It was liver gene expression done on an Affymetrix array. When you see RU1, that's a code link array, and BM stands for bone marrow, so there's a whole bunch of different tissues in here. And so what it tells you here is we use there's a p-value statistic, a Reddit score, then the severity score, and then it breaks down what all the animals are here. There was total animals 3 in this group. The normal incidence is 0. So how many had a normal incidence, how many have a minimal incidence, how many had a mild incidence, and then moderate or marked. And so as you can see here, as you go down the table, more of them had normal, a couple of them, and then you start seeing minimals and things along those lines. But these are all the experiments that caused, the chemical treatments that caused this phenotype here. And there's extensive documentation to explain all of these statistics and things along those lines. And you also have, when you go into expression studies, this is the entire study. So did a chemical at any time point in a given study produce this phenotype? And so, and you can, if you need to, you can export these. And so just go here, export, and it'll export as an Excel spreadsheet. Also, with the pathologies, it'll tell you what's how we describe the pathology. It gives you a couple of images and then gives you a description of what the pathology is. I should point out there is a help section, so if you go up here to help, click here, there's a reference manual, a tutorial. The tutorial is actually really good, and then associated data. There's the pathway legend, and then there's the different supported data formats, things along those lines, and descriptions of all the materials and methods for what was used to generate the data in the database. So we just looked under one domain, but you can look under as many domains as you want just to get a sense of what's in here. So you can, I went into compound domain and I search phenobarbital, and it gives you everything related to phenobarbital. You can click on one of these. I don't think methylphenobarbital was was evaluated in the database, but we've curated the chemical, and so it gives you all the chemicals that are structurally similar that were annotated in the database. Yeah, there were no gene expression studies with methylphenobarbital. There was with phenobarbital. And you go through these, so we'll just load phenobarbital here. Gives you the similar compounds, tells you what experiments caused, what genes were induced by phenobarbital. It's loading down here, so when you see it processing, that means it's going back to the server to pull in information and pull it up here. So this may take a minute. Git repressed, different experiments that phenobarbital was done in, so where gene expression was captured. So this is phenobarbital 5 days, liver, obviously it's going to be in rat. RU1 stands for the code link microarrays, and if you just go here, if you go here, that's RU1, then there's RG230, but you can see the different durations. This one's 3 days, this one's 5 days, this one's 1 day, that's the 1D, 3D, so that's how you read the names. There's a number of in vitro assays that were run as a part of the database. So it was a separate, the chemicals were given out to a separate company, and they ran them and returned the data. So it's kind of like Tox21 data a little bit. And so what it gives you is the results of the different bioactivities. So they had an assay targeting cyclooxygenase, vasoactive intestinal peptide, things along those lines. So it gives you a pharmacological profile. Then here it gives you the literature, tells you what was curated. Most of this is related to the target in vivo. Phenobarbital binds the gamma or the GABA receptor, and it gives you the curated activity potency. Tells you what the targets are. These are the gene targets. So the corresponding protein associated with this gene is what phenobarbital binds to. Motifs are a unique one. So there was an issue with interpreting a lot of the agnostically derived signatures. So what was done is they subsetted each of the they subsetted the genes into pathways and then built classifiers based upon just the genes in a pathway. And if the classifier worked, it was stored in the database. If it had some differentiation capacity, it was stored in the database, and now it's scored. And so with phenobarbital, one of the highest scoring motifs is actually it's a phenotypic signature that when you use the thyroid hormone synthesis and regulation pathway, the model shows up as significant. In this case with phenobarbital, as an SP score. There's again, to see this, all the scores and what the description of all the scores are for a given signature, you go up here to the help section, and then it shows you if you want to look at the pathway. So there's a whole bunch of pathways. So I just clicked here. So again, motifs, what they are is they said, okay, we're not going to use the entire set of gene expression data. We're just going to use a pathway set of genes that were measured on the microarrays, and we're going to build a signature. And if that signature had any predictive ability, it was deposited in a database, and now you can score against it. So this is the equivalent of an elevated serum alanine aminotransferase signature based upon thyroid hormone signaling. And if you open up this, you'll see it here. So the genes just in the thyroid hormone signaling pathway. There's one for BCR-ABL signaling, things along those lines. So these are just, in essence, supervised gene sets that were fed into the classifier to build the classifier. SPLP are more traditional signatures that are derived straight up by machine learning approaches. So basically you have a whole bunch of animals that have a given phenotype, a whole bunch that don't, and then you ask what genes are present, what genes are differentially expressed in that comparison. You build a model around that, and it's deposited here. Within this area here, you can actually look at all the models if you want to. If you go to signature here, so you select signature within domain, and so basically what these are is these are signatures that derive from the data, and they sit in the They sit in the database, and whenever you load gene expression data, they will then be scored against your chemical or your gene expression set. So in this case, what this is it's scoring against all the different phenobarbital treatments. And so you can see here the highest scoring one is neutrophilia. Next one is toxic DNA alkylator, but that's actually on the when you use the code link microarrays. So you see these are all specific to different microarrays. And then T rank is just another form of a signature that actually just looks at the genes that are basically most robustly affected within the differential in the model development. So these are just again a different way of deriving a gene set. So the genes in these signatures tend to be more robustly differentially expressed, but they may not be as differentiating between the phenotypes. And they might not discriminate as well between, say, hepatic hypertrophy and hepatic lipid accumulation. So you'll notice there will be a number of genes that are actually redundant across these signatures, shared quite extensively. So what about hepatic steatosis? Okay, so what do we have in here related to hepatic steatosis? I already showed you the pathology search. So you can go down there and go to histopathology and put in lipid, hit display, and then you can select one of these, whatever pathology you want to look at, and it would populate and tell you what the treatments produced it, what the frequency was, doses, durations, what microarray data is available, things along those lines. If there are specific chemicals you want to look at, I showed you that already. So carbon tetrachloride seemed to produce pretty robust steatosis already. That was one of the ones that was scored in the histopathology section as being the animals were most affected for lipid accumulation. I forget which one that was. But say you wanted to, one of the things I did find out is that if you go here and you go to pathway. So there's a bunch of unique pathways that are specific to DrugMatrix. So they actually curated this data themselves. It wasn't them just taking it from KEGG or some of the other IPA or things like that. So this is a rat-specific hepatic steatosis signature, or a pathway that they've put together. So you go here, you select pathway like I did here, and then type in steatosis. And what you can do is you can then load the steatosis pathway. And so what you have is all the genes that they've curated to that pathway, and they're here. If you want to, you can just export them if you want, export. And then you have all the compounds that have been annotated to the pathway. And then there are pathways that are, these are secondary pathways that are curated in the database that are associated with the steatosis pathway. And so you can see, if you just double click over here on the image, this is the outline of the pathway, and there's a whole hierarchy to how these things are all drawn to give you an idea of what how you end up in the end with hepatic triglyceride accumulation. So yeah, so there's a whole curated pathway, and it might actually give you an opportunity to take a look at some of actually, so no one's really written this up, so it's and it's all right here, and so it would be great if you want to do a review on it. And there's a whole bunch of this. So actually, all of the research on that went into creating this pathway is all curated here. So basically, you can just go out and grab all these things if you wanted to write a quick description of what's going on. You can do that. Also, there is a description here of how they created the pathway in the annotation, and pathway and all the annotations. So this can just be copied and pasted as a start point for evaluating mechanistic hepatic steatosis. Now, granted, some of these are pretty old, so you're going to want to probably update them. What else is there? Signatures. So there are, so if you go to signatures and you type in this lipid, it will display all the lipid accumulation signatures. And so there's specific identifiers and things like that. And so these are all data-derived signatures. There's the SPLP signatures, ASPLP. These were done for slightly different AFI arrays, but they're RG232. There are ones for, as you can see here, the CodeLink microarrays, and they're all versioned. We have other versions of these in the database that are not shown here, that are in the background, that we could give you too. But as you can see here, this one here is for hepatic lipid accumulation central lobular. This one's, and then you come down here, there's hepatic lipid accumulation macrovesicular, microvesicular, and you can open up any one of these. Let's just look at the macrovesicular here real quick. And right now, for some reason, there are no experiments aligning with that, which is, I think that might be a problem. And then, but there are the genes. And so what this tells you are what the genes are in the signature, and you can simply, if you want to work with these, you can simply export them and you'll get the AFI ID here. And then it tells you what chemicals were positive that were used to build the signature. So all of these produced this effect here to a level that was agreeable to build the signature. And what they did is they brought them in and they compared them to basically everything else. And it tells you that. So these are all the positive ones. And then it tells you how they made the positive class right here. It tells you what the RIT score was, and then the negative class was everything else in that organ, right? And then it tells you if there was anything excluded, and that gives you a label description. It tells you what the average true positive, true negative. You get the basic idea, basically the details on how well the signature performed, and then gives you a whole bunch of other information. Again, so you can pick any one of these if you wanted to. There's microvesicular. One of the things that they never did was actually go out and look at the different pathways, which was always always something that bothered me. But you can save these. You can save list as, I'm going to call it HLA MI central lobe liver. So that's RU1, so it's probably not the best, but we'll save that. And then what happens is it shows up over here in the workspace. So I basically save this gene list. And then what we can do, come on, I guess the save function. So you go over here, you can go to workspace, and there's all sorts of tools in the workspace. And so I can just go down the bottom here, that's a gene list. I can go to toolbar, and we can do pathway impact matrix, and we would have to do genes to pathways. That's what it was. Genes to pathways. Come on. And we just grab that and hit. We'll just call it microvesicular and hit translate. Now what we can do is we can come over here, grab this, grab it to the little display, and not surprisingly, you're getting a lot of inflammation sort of stuff. Acute phase response, P38, things like NF-kappa B signaling. And so yeah, so that's what we did is we just took the genes from the lipid accumulation microvesicular signature, and we just simply saved the list and saved the list here, and then we asked it to go take those genes and put them in the pathways, and then we simply dragged it down here to show you what pathways they were. So overall, pretty good. There are a number of other things that you can do in here. I just wanted to get you oriented to start off working with the interface. I'll make more videos to give you an idea of what else you can do. But again, the thing you probably want to do first is to actually look at the histopathology, and you can go to the study report. So that was one thing that I didn't show you. So there are actual reports here from the lab. So if you go to the expression study domain and just type in whatever you want, so we'll look at carbon tetrachloride. I'm not sure which one this was, but so what this tells you, so there's an expression study and there are expression experiments in the database. There's the expression study is when we've done multiple time points and things like that, so doses, so it captures, it's more just the overall chemical, whereas expression, in the expression domain, it's the individual specific expression experiment. So for this study here, which was done in 2001, you have many different expression studies, and they were done on either on the biochip is the code link arrays, and the gene chip or the Affy microarrays. But it shows you how many different tissues we looked at, how was the duration, what the dose, whether or not we designate high or low, percent of genes induced or repressed, total perturbed. But what it gives you it gives you all the histopathologies that were called. And so in this case, there was central lobular lipid accumulation in this study that was seen. And this, you remember this image from the previous visual. Then there is the path lab report. So if you click here on the path lab report, you bring this up. It actually, what it does is it tells you what all the parameters were for the clinical chemistry at different time points. So you can see here, shows you plots here, gives you the hematology results. These are all specific to the study. Shows you what the organ weights were, and then the animals each day, and it gives you the body weight effects. And then it gives you some canned images on what these are supposed to look like when you have inflammatory cell infiltrates. And these are all the pathologies that manifest, so you can see there were a lot in this case. And there's the lipid accumulation. So, but these are not specific to the study. They're simply canned canned images that are stored in the database. So I bounced around a little bit. The other thing that's really nice for the toxicologist is it actually gives you the low dose, high dose selection, like why it picked the certain doses. Typically the high array is the dose that was considered a 5-day maximum tolerated dose, and the low array is what's supposed to be pharmacologically active in the rodent. But that's a good basic overview, I think, and I think you just need to get in there and start trying things out, and then come back to me with questions. I'll show you some of the other stuff later on.